It's very nice to be online with all of you. I learned a lot of things from Ilya's talk, so it's always a pleasure to see the classics being revised, or not, but expanded. So I really appreciated this possibility of revising the classic biophysics. I'm gonna be talking about an escort complex that is actually working at the membrane, but I'm not going to go into the details of the lipids themselves, more about how they get remodeled by the complex. So you probably all know that the intraluminal vesicles that are formed inside the endosomes were the place where the escort pathway has been discovered by Scott Emmer through a genetic screen. And this is shown here. And the peculiar aspect of the formation of ILVs, which otherwise seems very similar to endocytosis, is that the deformation and the fission reaction to generate those ILVs is actually generated from proteins, in this case the escort-3 complex, that are situated in the cytosol, which means inside the bud instead of being outside the bud, as it is the case for endocytosis. From that on, the escort-3 complex has been actually involved in essentially all the pathways that require the cytoplasmic proteins to be inside the bud of membrane deformation instead of being outside. And this is true in many cases, as you can see here. I will just highlight a few of them. The very first one that was actually introduced by Wes Sundquist is actually the budding of HIV and the scission reaction that occurs there to liberate the virin is actually generated by or performed by escort-3. Now, another essential function, of course, is the final step of cell division, abscission, and this is again generated by escort-3, as you can see from this paper from Daniel Gerlich. And the third, okay, missed the third one, but essentially what I want to say is that this complex is actually the only one we know that has a remodeled function that works on every membrane: plasma membrane, nuclear membrane, endosomal fractions, autophagosome. And so it's a very essential complex of membrane remodeling. I'll go through a very simple structure, and I will simplify it by using the term layer and essentially 3 layers, even though this is not completely accurate, as you will see in the rest of the talk. The first layer is composed of VPS20 and SNF7. These are the yeast names. I won't use the mammalian names. And VPS20 is a nucleator, and SNF7 a main component that gives the structure to the complex. I'll show you that in a second. The second layer, VPS2 and VPS24, are binding to SNF7, and they recruit a third layer, which is D2-IST1. What we discovered with Daniel Gerlich is that the VPS4 molecule that uses ATP initially thought to disassemble the complex is actually here to trigger turnover of the subunits in cells and also in vitro. Now, why is this complex particularly interesting for biology? Is because it's the only remodeling complex that you find in archaea, and archaea are thought to be, and this is shown on this slide, are thought to be a transition from prokaryote to eukaryotes, and thus to be involved in the formation of the endomembrane system. This is a theoretical paper from Buzzbaum that maybe some of you have read, and that's a inside-out theory of eukaryote formation. And I want to strengthen that the first step is shown here. To go from A to B, you have to make protrusions that will connect the one prokaryote to another one, and in order to lead to the formation of more complex endomembrane system. And these protrusions that are postulated in this paper actually look very much like structures that have been seen by Phil Hansen when she overexpressed the human version of SNF7 in cells. And you see here that at the inner leaflet of the membrane, you have this dramatic formation of spirals. And these spirals can lead to the formation of tubules that are protruding out like philopodia coming out of the cell, as you can see here, when you inhibit VPS4 on top of overexpressing SNF7. So this led to the hypothesis, together with Martin, we talked about it a long time ago, and he wrote a theoretical paper about this hypothesis, the possibility that these spirals could actually accumulate elastic stress and deform the membrane by buckling, and that's shown here. You essentially need filaments that are elastic, curved, so they would form spirals, and the driving energy would be the polymerizing energy of the SNF filaments. So in my lab, to test this hypothesis, Nicola started to work on supported bilayer, and you see this in gray here, reconstituting the SNF7 protein, so the layer 1 that forms patches, and these patches turned out to be made of many spirals, as you can see on this AFM picture. We can actually look at them growing, and that's the chymograph that you can see. This is not a single spiral, this is a patch, so it's full of many spirals. And we could look at those growing spirals by high-speed AFM with Lorena and Simon, who are experts in the high-speed AFM. And from these data, what we could postulate is the mechanism that is shown on this animation, which is spiral spring buckling. So if you have a filament that grows into a curved filament at the surface of a flat membrane and forms a spiral, this will not spontaneously accumulate stress because the filaments in the center are overbent and the ones outside are underbent. And to release this stress, one easy process is buckling that you can see now. And this buckling will form a protrusion that looks very much like the protrusion seen by Phil Hansen. Okay, now we had one problem in this study is that we never observed cylindrical membrane structures with only SNF7 alone. And we had a hint. We had a hint from Mike Heaney's paper when he was with Scott Emmer as a postdoc, because they saw a transformation of spiral into helices only when they added VPS24 and VPS2 to the media, which are actually the second layer, components of the second layer. So essentially what we're saying is that the buckling transition could be transformed, could be generated by adding the second layer. Is that true? Well, we knew that adding VPS2 and VPS24 to SNF7 spiral would thicken them. And you can see that this is a spiral made of SNF7 alone, and this is a spiral made of layer 1 and layer 2. And the hypothesis here is that we form a secondary filament on top of the initial filament made by layer 1. Now, Joachim in the lab went a bit further into testing the possibility that adding the second layer would trigger the buckling transition. And indeed, what he observed was that you get cylindrical structure with membrane inside, circled with the filaments, as you can see on this nice 3D reconstruction done by cryotomograms. The most striking feature of those is actually that you don't get a pure cylinder, but you actually get a helical tube, as you can see from the left picture here, and from, of course, the tomograms. So we wondered how this could be possible. And he went a bit further in analyzing the components of these filaments, and he found out that there was essentially two classes of filament. The equatorial one that were circling from the outside the filaments, and the polar one that were essentially binding to above and below the helical face of these filaments. And these filaments are very similar in thickness and dimensions then to the filaments we had seen on flat supported bilayer. So one of the problems here was that the geometry of this structure actually forces us to accept the idea that the polar filament and the equatorial filament, if they are the same, should bind the membrane with a different orientation, as you can see from the drawings here. And that is just to say that the equatorial should bind inwards and the polar should bind on the top, so with a 90-degree angle to the membrane. If this is true, we should be able to observe that. Fortunately, Joachim observed that putting detergent in the buffer helped into forming structures with, at least at the beginning, seem to be devoid of membrane, and that you can see in the A and in the B. And fortunately, he had several types of structure, one looking very much like the polar and one looking very much like the equatorial filaments. And he managed, with the help of Adam Frost, to get a high resolution structure of the polar form. And what he found was that actually the orientation of the monomer in this case, as you can see on the left, is actually 90 degrees perpendicular to what was postulated by the structure made by Adam a few years ago, where you can see the binding part as a blue color. So you can see that the orientation is 90 degrees. The arrows will show you how the membrane is actually binding in our structure here on the right. So indeed, these filaments can bind with different orientation, and that explains part of the structure. So to really understand the stability of this helical tube, which should actually not be stable for energetic reasons, we went back to Martin and Luca, and they made a theory to explain how this could be possible. And you have here a short drawing with the three filaments that are on the structure on the left, and you have the shape diagram and the stability of this. And you can see that you have a large area where only the straight tubes should be available, but you also have an area where you can have helical structures following the things. And from this theoretical analysis, we can understand essentially three ingredients that are essential, for the stability. First, that you have to have a helical scaffold. You have to have a high bending and twisting rigidity of that scaffold. And the membrane should bind to the filaments with different orientations and different affinities in both equatorial and polar configurations. So what we're getting out of this is that the addition of VPS2 and 24 rigidifies the SNF7 spirals, and that could also give an additional twist to the structure, driving the buckling. Of course, that means that the escorts have unique features, in particular the possibility to bind at least in two orientations, and with one of them being favored in terms of affinity with the lipids. We went on testing this hypothesis. And in particular, we wanted to test whether buckling could be triggered by the addition of VPS2 and 24. That was the work of Anna in the lab, a PhD student. And I just want to mention that she just got the Birnsteel International Award a few weeks ago, which is a great European award for great achievements for PhD students. So what she worked on was to understand how the potential sequential addition of subunits could trigger the buckling transition. So she used the same assay than Nicola, as you can see here, so supported by layer, and she looked at how molecules, subunits coming further into the complex would bind to the initial SNF7 molecule patches. And what she could find is that there's really an order of sequencing in recruitment. So I'm going to go through rapidly. SNF7 essentially recruits VPS24 and VPS2, so layer 2, which itself recruits or can recruit D2, which binds to VPS2, and further recruits IST1, which is a component of layer 3. And amazingly enough, she found out that the binding kinetics were exactly matching this one-to-one binding affinity. So that suggested that the protein could actually be recruited sequentially. Now she looked at how they were depolymerized from the complex using VPS4, which I repeat is an ATPase that should trigger disassembly. And so what she did here was to assemble all proteins onto the patch, add VPS4, and look what's the dynamic of disassembly of those proteins. And I think it's very clear from those data that you have a clear sequence in depolymerization, first SNF7, followed by VPS24, VPS2, D2, and IST1. And so again, layer 1 would go off before layer 2, which would go off before layer 3. So she went on trying to analyze this by essentially putting all proteins at the same time and seeing whether there was a recruitment sequence and a depolymerizing sequence that would organize time with space. And that's exactly what she did. I'm not going to go into the details of this. She analyzed one protein with using SNF7 as a reference, and she can now plot the dynamics of each protein all together. So all proteins are added at the same time, and she looks at one protein, and you can see that they come into peaks one after the other. So we start with SNF7, which forms the initial patch, and then SNF7 peaks, followed by VPS24, and then VPS2, and then D2 and IST1 to end up. So that organizes a sequence of recruitment and depolymerization in vitro, which I think is a beautiful example of dynamic waves of recruitment spontaneously organized by affinity and ATP. So what I'm telling you here is that SNF7 recruits VPS24 and VPS2, and VPS2 has a pivotal role in changing layer 2 to layer 3 by allowing the exchange of 24 to D2, and D2 itself recruits IST1 while the layer 1 is going on, and we are left with D2 and IST1, the final complex at the end. Why is this interesting? Because this complex stands for longer, as you can see from these images. If you add all proteins and add VPS4, 30 minutes after, the red dots are VPS2, IST1, D2 left, and you can see that they form protrusions with time that seem to be going inside the vesicle, and this is done on giant liposomes. Now, if you look by EM at these structures, and I'm going to go fast on this, you can see that the vesicles are deformed with inward protrusions made by the spirals, as seen by Phil Hansen on the cells. Now, if this is the intermediate stage, so that's when you have layer 2, and if you go to layer 3, the protrusions are getting even larger and longer, but smaller, more constricted, and that's the transition we see here. So to resume, what we are saying here is that the layer 1 will be exchanged with layer 2, which finally will be exchanged with layer 3, and this is all driven by ATP consumption by VPS4. How can we explain this? So while we were revising this paper, a paper from the lab of Andela Sarik, who was using simulations to study the remodeling aspects of membrane, actually came up with a very nice study which proposed that twisting or tilting the axis of the filament could be sufficient to drive deformation. And you can see here what's happening. If you have a filament composed of 3 filaments, protofilaments, and you just tilt those protofilaments, you go from a flat state to a curved state. Of course, that's not completely coherent with what we have observed. So we discussed with Andela, Buzz, and Sharon whether having 2 filaments that sequentially arrive to the membrane and bind to the membrane, one being with a certain orientation and the other one with a 90-degree tilt, this could trigger the same kind of transition, which is what we observed. So if you start with a flat spiral, add the second layer, which has an orientation to bind to the membrane of a 90-degree, then you will form a shallow pit, which we observed, and then removing the layer 1 will form these deeply invaginated structures that we also observed. And that's very consistent with what we saw. Now, to verify the possibility of the twist addition during the remodeling of the SCOR3 complex, Joachim was kind enough to look in his samples with SNF7, VPS2, and DIT2, so this, and to compare them to the previous study he had done on the tubular, the helical tubes that he had observed. And you can see that I hope it's clear from these 2D class averages that the filaments actually have additional twist when you put D2 as compared to the initial VPS2, VPS24. And that's the drawings that you can imagine from there. So indeed, we think that exchanging VPS24 to D2, so essentially moving from layer 2 to layer 3, adds up a twisting component to the helical tube, and that drives deformation. So what's going on? That's a small animation done from Nicola in Andela's lab. And so initially you have a shallow pit or almost flat structure with two components, two filaments, and one is being removed by addition or consumption of ATP, and that drives a deep invagination of the membrane. The third layer is actually also constricting, and you can see on those pictures that we get very, very tiny invagination if we add IST1 to D2 VPS2 structures. This is interesting because the size of those is actually, those structures was actually already studied by Adam Frost, and it has the dimension of a dynamine 1 complex, which is a fission complex. So we wanted to know whether this could be the fission mechanism and whether the addition of the D2 IST1 complex would fission membrane. So we just added VPS2 D2 to vesicles, and that created tubules that have somehow constricted necks. And the addition of IST1 did not really trigger a difference in shape, a difference in shape. I hope you can see from here and here the helical color of VPS2 D2. But if we activated the exchange or the removal of VPS2 and the addition of IST1 using VPS4 and ATP, then we saw very constricted necks, as you can see here, some of them not being constricted at all. And if we looked at the average diameter instead, indeed, we went from 20 to 10 nanometer, which is consistent with fission dimension as known from the dynamine field. Now, also comparing to dynamine, we knew that if tubules are attached to the surface, they spontaneously break under constriction. And so we pre-adsorbed the VPS2 D2 IST1 tubes to the grids and then treated them with VPS4 ATP. And in this case, we saw fission, as you can see with this alignment of vesicles that have been separated during the ATP treatment. And so what we're saying here is that if we add VPS4 and ATP, this triggers the exchange of VPS2 with IST1, and we are left with a more constricted D2 IST1 complex that can trigger fission. That's shown here. Now, what I've shown you is, of course, on the wrong orientation. This is from outside, and the question was, can we get fission in the correct orientation, meaning having the escort complex inside the neck of the membrane and not outside? So Anna went on creating this artificial budding situation or artificial endosome by using silica beads that were spontaneously endocytosed by giant liposomes, forming next to which the escort3 complex could be added from outside in the buffer. And with this, she was able to form these vesicles that you see, the membrane is shown in blue, on which the escort complex was specifically binding to the neck of beads that you see a bit more intense in blue. And she could reproduce the entire sequence of recruitment that she had observed on flat membrane before. Now, doing that, she was able to test whether this sequence would lead to fission by just using an optical trap and taking any of these beads and pulling them inside. And if the bead, once pulled, was released, meaning diffusing freely, that could mean that the fission had occurred during the escort3 treatment. And I want to strengthen that she did many tests with subcomposition, and none of them worked with the exception of adding all the sequence together. So all the proteins are added together, SNF7, VPS2, 24, D2, IST1, and of course VPS4 and ATP. And in this case, she got up to 75% of beads that would break. So that's really the fission mechanism in this time in the correct orientation. So that's the mechanism that we have postulated. And I hope it gives a little bit of understanding of how SCOR3 complex works. And I'm just going to show you a preliminary animation from Mandela, Sharon, and Buzz that tries to recapitulate the entire mechanism. So initially you start with essentially the 3 layers, and if you remove layer 1, you will form a deep invagination, as you can see from here. Now, if this invagination can go on, it will get constricted more when you remove layer 2, and this will lead to fission eventually when it breaks. And that's what we're working on, and we think we have at least a global mechanism of how it works. Of course, the details have to be around. I think I'm going to stop here. I had a little bit of more slides on how membrane tension regulates the process, but we should definitely stop here. And I'm open for a few questions if you have. And I'm going to just show the very last slide here to thank people. Again, I would like to thank Vincent Mercier. I had no time to show his work, Joachim, and of course, Anna, which most of the work I've shown today was from her PhD studies. Thank you.